it's my pleasure to introduce Dr. Colak. She's an assistant professor at the Brain and Mind Institute. She earned her PhD from the Ludwig Maximilian University in Munich, in Germany, and has done a postdoc in New York, exploring the role of local mRNA translation. She also studied RNA degradation, the nonsense mediating mRNA decay, NMD, as a mechanism for exon guidance in vivo. She has made like several discoveries, including one of I consider like a seminal paper showing that the fMRI1 gene, the one linked to Fragile X, is regulated during embryonic stem cells due to the expansion of the repeats, and the mRNA actually interacts with the DNA that triggers the expression of the gene, explaining why sometimes you don't see that expression. So that was one of the first really important mechanisms that you see, that a no-coding RNA could actually bind to the DNA and regulate gene expression in a very important gene. So she has lots of publications, which is quite amazing. And we recently exchanged some emails, and then she pointed out a recent paper on astrocytes and autism. And I missed that. I was embarrassed, and I was reading. I said, "Oh my gosh, this is amazing! A tour de force." And I said, "Oh, I have to invite her because we do have a strong gliobiology here in Southern California that would appreciate her work." I was familiarized with her work on brain organoids and schizophrenia, recent work that was also like fantastic. But this one on autism is one of my favorites. So I invite her and thank you for accepting the invitation in such a short time frame. And as soon as the announcement start to get out, I mean, I received lots of email from colleagues saying, "Oh, thanks so much for inviting her. This work is amazing. I can't wait to see it." And I'll just read like the email from Miles Wilkinson, who's probably here in the audience. And he said, "Alison, Dilek is terrific. She did tremendous work with Sammy Jeffrey on both in MDA and Fragile X. Both its Cell and Science papers." And then, I mean, she mentioned that she got to know you and Sammy and wrote like Nature reviews on MDA and the brain. And she's following your career. So he's a big fan as well. So I'm so happy that you accepted, and I can't wait to share my excitement of your recent discoveries here with my colleagues in Southern California. Thanks so much, Dilek, and the floor is yours.
Thank you very much for invitation and also for this kind introduction, and special hello to Miles. Actually, I'm supposed to write him an email because we have more exciting data on NMD, so I need some advice from him, and I have to share our new data with him. So it's my pleasure to give the talk today. I'm very happy to be here. As Alison mentioned, in my lab, we are interested in understanding molecular and cellular mechanisms regulating synaptic plasticity, both in a healthy state and a disordered state. And we are particularly, we are focusing on regulation of RNA and its contribution and glial contribution to neurodevelopmental and neuropsychiatric diseases. In our recent study, we particularly focused on astrocyte pathology and astrocyte dysfunction in autism. So I'm sure, you know, all the researchers know about autism, but, you know, just a brief introduction. Autism and ASD are both general terms for a group of complex disorders of brain development. Autism affects millions of people worldwide, and approximately 95% of autism diagnoses are idiopathic or non-syndromic. In other words, they are not associated with known genetic anomaly. There's no cure for ASD, and the reason for that is because we have not fully understood molecular and cellular mechanisms underlying this disease. Having said that, it's not that we don't know anything about it, because genome-wide association studies and animal model studies from syndromic ASD, such as Fragile X syndrome and Rett syndrome, have suggested that dysregulated synaptic function accounts for most of the abnormalities in this disease. And interestingly, the onset of ASD symptoms coincides with activity-dependent synaptic plasticity events. So synaptic plasticity, again, I'm sure most of you are familiar with that term, but in basic terms, it means it refers to the ability of a synapse to become stronger or weaker, because synaptic transmissions between neurons can be either enhanced through long-term potentiation, or they can be depressed through long-term depression. And in synaptic plasticity, both LTP and LTD are considered to be the foundation of learning and memory, because the majority of the excitatory synaptic contacts occur on dendritic spine. Spine morphology and spine densities are also very critical for synaptic plasticity and in turn for learning and memory. And interestingly, ASD postmortem brains also exhibit aberrant spine densities in various brain regions. So why were we interested in astrocytes at the first place? Because astrocytes are integral components of the tripartite synapse. In a single human astrocyte can contact up to 2 million synapses, and it's around 100,000 in mouse brain, I think. And astrocytes have been implicated in the regulation of synapse formation, secretion of factors that affect dendritic arborization, synapse elimination, and removal of excess neurotransmitters to prevent excitotoxicity. And one of them is, the most famous one is glutamate. And especially astrocytes have been shown to positively regulate LTP, and proper astrocyte function is required for memory consolidation particularly. So what do we know about astrocytes and ASD so far? So co-culture experiments revealed that the presence of astrocytes derived from the brains of syndromic cases such as Rett syndrome or Fragile X syndrome altered synaptic transmission in healthy neurons. And transcriptomic and immunohistochemistry studies in postmortem tissues also found that there was astrocyte reactivity in the cortices of individuals in brain cortices of individuals with ASD. So reactive astrocytes are astrocytes that undergo molecular, functional, and morphological changes in response to pathology such as injuries or disease. Despite these implications, the precise role of astrocytes in synaptic abnormalities observed in autism is unknown. And we even don't know if this pathology we see in postmortem brains, this reactive astrocyte pathology, is primary or secondary to the disease. So to study astrocyte pathology, we use patient-derived stem cell models. This also doesn't need an introduction because animal studies have been helpful to understand certain aspects of autism, but they are not sufficient to fully capture the disease pathology. And it's a brain disease, and brain makes us different than animals. And also postmortem studies have limited insight into understanding the onset and progression of the disease. So currently, patient-derived stem cells and patient-derived cells provide an ideal experimental system to understand molecular and cellular mechanisms underlying polygenic diseases such as autism, because it provides us the opportunity to study living patient cells in a dish. And 3D brain organoid technique actually moved iPSC culturing also a huge step forward. As you all know, they're considered to be mini brains in a dish. It's a 3D environment, and it recapitulates various brain regions. But for me, one of the most important aspects of organoid technique is that cerebral organoids recapitulate the temporal sequence of cortical development seen in developing embryos. Namely, neurons are generated first, then glial cells are generated. So we exploited this organoid technique to study astrocytes, ASD astrocytes. Basically, what we did was we obtained stem cell lines, iPSC lines from 9 healthy controls. And we have obtained 10 independent iPSC lines from 10 ASD patients. And we generated organoids from all of these lines. So in these organoids, like as I said, like neurons are generated first, then astrocytes are generated. Astrocytes appear usually around day 60. And at day 70, 75, we dissociated these organoids and selected for astrocytes, enriched for astrocyte by using a media that selects for astrocytes. So we basically used organoids only to populate a healthy and diseased astrocytes. So we could choose a protocol, a 2D differentiation protocol to generate astrocytes, but we wanted to generate the astrocytes as spontaneous as possible so that they would carry disease-specific features. Because, you know, so 2D differentiation protocols might use, growth factors, some other, excessive amount of like factors or sometimes FPS that could alter disease-specific features in these cells. So we didn't want perfect astrocytes. We wanted to have disease astrocytes. And most importantly, the organoid system also allows to populate ASD astrocytes in an environment that mimics the early ASD brain pathology, meaning presence of ASD neurons. So we then labeled these astrocytes. We infected them with a virus expressing GFP and transplanted into the brains of mice at postnatal stage, and then studied, addressed whether these ASD astrocytes induce any synaptic plasticity or synaptic plasticity deficits or behavioral alterations in adult chimeric mice. So we basically generated a humanized mouse model of ASD, chimeric mice. But before I go into these, transplantation experiments and behavioral or synaptic plasticity experiments, I want to show you the characterization of our organoid-derived astrocytes. I will also show you an experiment where we attempted to understand, to address the inherent deficits in these astrocytes, and I will come to that in a couple of minutes. So this is how our, typical organoid cultures look like. This is how, this is our organoid around day 75. This is the time when we dissociate our organoids to isolate astrocytes, and this is how control and ASD. These are representative organoid images from both control and ASD group. They are similar in size at the time of dissociation. And the first thing we did was, of course, to examine if they express astrocyte markers. They express various astrocyte markers. The well-known ones are ALDH1L1 and GFAP, but they also express AquaP4, vimentin, and S100 beta. So we also wanted to assess the reactivity status of these astrocytes, because I mentioned during the introduction that in ASD postmortem brains, astrocytes show reactive status. So one of the most reliable markers of astrocyte reactivity is F-actin upregulation. F-actin basically refers to actin filaments, and because astrocytes undergo morphological changes in reactive states, F-actin is drastically upregulated. You can follow F-actin with a phalloidin peptide that selectively, the specific labels recognizes F-actin. So we did phalloidin staining in our astrocyte cultures, and here are representative images like from 3 control lines and 3 ASD lines. And this is on the left side is our regular cultures with 2% FPS, minimal FPS, and we had to we needed a positive control to compare our astrocytes. Like we needed astrocytes that were reactive. And it's known that high levels of serum can induce reactivity in astrocytes. That's why we also cultured our cells in 20% FPS that, as you can see, resulted in a significantly higher F-actin signal compared to our regular cultures. And these are quantifications here. So if this culture is considered on the right side, 20% FPS is considered like reactive astrocytes, we concluded that our organoid-derived astrocytes are not overtly in reactive state, as they had very weak, low level of F-actin staining. So to determine, to identify inherent pathological deficits in these cells, we took an unbiased approach and compared the protein profiles of control lines, compared the protein profiles of ASD lines with control lines using tandem mass tech liquid chromatography mass spectrometry. So here, there are two independent experiments are shown here based on the availability of the lines at the time, but both experiments showed high reproducibility and the identity of the proteins that were detected were almost identical between the groups. So interestingly, we found a lot of differentially regulated proteins in ASD lines when compared to control lines. However, those differentially regulated proteins were not common across all ASD lines. This is expected because this is a non-syndromic ASD, this is an idiopathic ASD, and probably each patient harbors different genetic variants that result in varied protein expression. However, using the same data set, we did an analysis called ingenuity pathway analysis. So this analysis identifies upstream and downstream predictors. So it determines if varied protein combinations across different samples converge on similar biological pathways. And indeed, this analysis determined that varied protein expressions across different samples converged on calcium signaling in ASD lines. So most of the proteins that were differentially regulated in ASD lines, although they were different proteins, they were all linked to a calcium pathway. So what do we know about calcium signaling in astrocytes? So calcium signaling in astrocytes is very important. It's very critical because astrocytes respond to neuronal activity via elevated cytosolic calcium concentrations. In baseline conditions, there is, some calcium activity going on in cytoplasm. There is certain levels of calcium, but most of the calcium is stored in endoplasmic reticulum. And upon stimulation, it's called evoked calcium response, increases of astrocytic calcium, so the release of calcium from endoplasmic reticulum to cytoplasm, lead to release of glia transmitters from astrocytes, including glutamate, ATP, and GABA, that in turn controls regulate synaptic transmission and modulate synaptic plasticity. So to confirm this predicted deficit in ASD astrocytes, so far it's a predicted deficit, we measured calcium activity using 2-photon live cell imaging. We loaded our astrocytes, controlling ASD astrocytes with a calcium sensor dye, fluo4, which excites at 488 and can be measured. You can see here, these are still images taken from 2-photon videos that at baseline, there is calcium activity, but upon stimulation with ATP, in this case, the activity is increased, and you can see that in the heat map plots too. So, however, this increase in response was much higher in ASD astrocytes compared to control astrocytes. So both groups, like both cells, responded to stimulation by with an increase in calcium, but the increase was much higher in ASD astrocytes. So when we discuss this, so we are not really calcium experts, but when we discuss this with calcium expert people, especially in astrocytes, they told us that in astrocyte, calcium signaling, calcium activity, both in cell soma and in the processes of astrocytes actually contribute to calcium signaling in these cells. And because astrocytes acquire their complex and large morphology in vivo, we can capture the real calcium phenotype in vivo. So, if we didn't do these experiments in vivo, we would not be really sure whether there is this calcium deficit in ASD astrocytes. So this brought us back to our transplantation experiments, because we said that, okay, we are going to put these cells in an in vivo environment. Maybe before we do our behavioral and synaptic plasticity experiments, you know, we could record calcium activity in transplanted human astrocytes. So going back to that scheme again, we generated organoids from control lines and ASD lines, and we selected for astrocytes, and we labeled these astrocytes and transplanted them into immunocompromised mice at postnatal stage. So postnatal stage is critical because, especially postnatal day 2 to 4, because it's when astrocyte generation peaks. So the brain allows generation, migration, and maturation of astrocytes. So it's the perfect time point for astrocyte development. So this is how chimeric brains look like. These are what you see in green is the GFP, our labeled astrocytes, and these are sagittal sections at different mediolateral positions. And you can see that organoid-derived astrocytes, transplanted astrocyte, migrated throughout the cortex and survived into adulthood. And here in the lower panels, you see higher magnifications from same sections that these cells populated, especially like cortex and hippocampus. So yeah, I mean, in addition to GFP, we also wanted to confirm that these cells are human cells, human astrocytes. We used an antibody that specifically recognizes human GFAP epitope, and so that staining also confirmed that these human transplanted cells survived in the adult brain and populated cortex and hippocampus. And we also examined if these cells retained their astrocyte identity upon maturation in vivo, and stained, co-labeled these sections with GFP and ALDH1L1 or GFAP. And here quantifications show that more than 90% of the GFP positive cells were either co-localized with ALDH1L1 or GFAP. So transplanted astrocytes retained their identities upon maturation in the adult mouse brain. So most importantly, these cells did not differentiate into neurons. So we examined more than 800 cells across several chimeric brains, different iPSC lines, that there was no single co-localization between GFP and neuronal marker NOI. And morphology-wise, also you'll see, you know, various different pictures that they really did not look like neurons. To validate our the calcium deficit phenotype we observed in vitro, so we measured calcium activity in live mice, actively responding to the environment using 2-photon calcium imaging. And for this, we infected our astrocytes before transplantation with a virus expressing calcium, genetically encoded calcium indicator GCAMP. GCAMP is the fusion of calmodulin and GFP, and you know it fluoresces when it binds to calcium. And we transplanted these astrocyte expressing GCAMP again into the brains of mice at postnatal stage. So the first step in vivo of 2-photon calcium imaging is placement of a cranial window on top of the somatosensory cortex. And when I say window, I literally mean window, because this is a 3-millimeter glass window. And after that, we let the mice to recover for several weeks. So these experiments were done. We were very lucky because our neighbor lab is the lab of Connor Liston. I mean, literally neighbor, and his door is his room is just behind this door. So they're experts in calcium signaling, and you would think that it doesn't get better than that, but it did, because his senior postdoc Ben Huang has specific expertise or experience in calcium imaging in astrocytes. So he performed these experiments. And this is how the setup looks like. You see a mouse under the objective, and the mouse is on a floating platform. That's critical because floating platform prevents image motion artifacts. And because we were interested in capturing calcium response upon stimulation, so we were interested in this evoked calcium responses, we had to stimulate this mouse as well. So here, and we did that with air puff application to face of mice. So you see a tube here, it's called air puff tube, and each time when a mouse received air puff to the face, it startled. And then Ben measured startle-induced changes in calcium transit. You can see here that this is baseline calcium here, and upon startle, there is a response to startle, there is increased calcium levels. And here, this is an astrocyte, a transplanted astrocyte in vivo expressing the calcium sensor GCAM. So the transplanted human astrocytes displayed a variety of calcium responses to startle behavior. So it was, of course, more complicated than in vitro. So for example, the first group resembled our in vitro astrocyte group. So upon startle, they showed an increased calcium response, but you can see that the response was much higher in ASD astrocytes. So another group showed a decreased calcium response to startle behavior. But in that decrease, even though the total levels seemed similar, or the decreased response was similar, but the fluctuations in calcium was much higher in ASD astrocytes. The third group responded to startle behavior with no change in calcium in cytoplasm. But even in that case, there were increased fluctuations when compared in ASD astrocytes when compared to control astrocyte. So this in vivo experiment, especially this first group, confirmed that there is an aberrant calcium activity in ASD astrocytes. So we were already interested in addressing the role of these cells or the contribution of these cells to synaptic plasticity deficits or behavioral abnormalities seen in ASD, and we would test that in chimeric mice. But this further raised the question, is this phenotype reflected to ASD hallmark behavior as well as learning and cognition in chimeric mice? So before doing any specific behavioral experiments, we first wanted to assess general motor activity of our chimeric mice. And to do this, we used this well-known test called open field test. In this test, you place mice in a special box. This box has photo beams, and the software tracks beam breaks in XYZ planes. So therefore, it accurately measures general motor activity or locomotion of mice. But in addition to assessing or recording general locomotion, this software also helps us to have an idea or to interpret anxiety type of behavior in mice, because it can measure center preference score by recording the time, by tracking the time the mouse spends in center zone versus edges. For example, an anxious mouse, the center preference score of an anxious mouse would be lower than the center preference score of a healthy or no anxious mouse. So in this test, both chimeric groups, control chimeric mice and ASD astrocyte chimeric mice performed similarly. So this showed that the injection protocol did not affect ambulatory activity and is not a confounding factor in the interpretation of other behavioral results. But more importantly, that ASD chimeric mice did not show altered locomotion activity or anxious activities, anxiety compared to control mice. So finally, we come to ASD hallmark behaviors. ASD diagnosis is based on 3 categories of behavioral criteria, as you might know: abnormal social interactions, communication deficits, and repetitive behaviors. To score sociability, we used, again, a well-known a test called 3-chamber social interaction test. So in this test, you place mouse in a box with 3 chambers, In the habituation phase, the test consists of two phases. In the habituation phase, the first phase, you place mouse in the middle box, and the mouse can freely walk between compartments because they, they are open. But you place objects, usually a wired cup, a meshed cup, in ant chambers. At the end of habituation phase, you remove the object from one of the ant chambers, and you replace the object, the cup, with a stimulus mouse. So in the social preference phase, this chamber where you place the stimulus mouse is considered social zone, and the chamber that still contains the object is considered non-social zone. In the software, and you let mice again to explore your test mice to explore different chambers for 10-15 minutes, and the software then tracks the time the mouse spends in social zone and non-social zone. So in this experiment, again, both groups performed similarly. So ASD, astrocyte chimeric mice, did not show deficits in sociability or in social interaction. So to test repetitive behavior, to assess repetitive behavior, we used, again, a commonly used test, a marble burying test. Mice, they like burying marbles, burying stuff in general, but also marbles. So we placed 20 marbles in each cage and let mice bury these marbles in a certain time. And at the end of the test, we quantified the number of the marbles that were buried, and we considered a marble buried if 60% of the marble was covered. In this test, ASD chimeric mice behaved significantly different than control mice. They buried way more marbles, as well as showed increased digging behavior. So these two tests concluded that ASD astrocyte chimeric mice exhibit repetitive behavior, but intact sociability. So because astrocyte function is critical for learning and memory, and because I have to tell this now, if I didn't tell earlier, that all of our ASD lines come from individuals with low IQs. We also wanted to assess if ASD astrocytes induce any learning or memory deficits, and we phenotype the mice for different types of learning and different type of memory. So for memory, for fear memory, or fear memory, we used a classical fear conditioning test. Basically, in this test, you pair a conditioned stimulus, which is an audible tone, with an unconditioned stimulus, which is a mild food shock. So you teach the mouse to learn that the audible tone is associated with the mild food shock, because each time there is a tone, right after that, there is a mild food shock. So on day first, you do 3 trials and you teach mice to be fearful of the audible tone. And when a mouse is fearful, it instinctively freezes. So you measure the freezing behavior and use it as an index of mouse's ability to learn the task. So at the end of day 1, at the end of the trials, this is how the freezing behavior looked like, and ASD chimeric mice and control chimeric mice performed very similarly. So ASD chimeric mice did not show deficits in associative learning. So the next day, you bring the same mouse to the same cage, the same context, but this time you don't apply audible tone, or there is no food shock. This time you again measure freezing behavior, but use as an index of mouse's ability to recall the contextual memory. The greater freezes, the greater memory. So in this contextual memory task, ASD astrocyte chimeric mice performed differently than control chimeric mice. They showed attenuated contextual fear memory. On day 3, you take the same mouse, this time put in a different chamber, it's a novel context, and you only apply the tone, and you again measure the freezing behavior, but this time you use it as an index of the mouse's ability to recall the cue memory. ASD chimeric mice did not show deficits in cue memory either, similar to associative learning. This experiment concluded that ASD astrocyte chimeric mice exhibit impaired fear memory. So we also assessed spatial learning and spatial memory using Morris-Wader-Maze test. In this test, you basically place mouse in a swimming pool. It's an opaque pool, and you hit a platform. You put a platform submerged in water, and you teach mouse with distal cues on day one to find the hidden platform. They are willing to learn. They are willing to find a hidden platform because they don't like swimming. Similar to fear conditioning test, there are several trials. Actually, they have 4 trials for 4 days. It's a difficult task. And eventually, at the end of all these trials, it takes mice much less time to find the hidden platform because they learn. And in this task, both groups performed similarly. ASD astrocyte chimeric mice did not show a deficit in spatial learning. So on the 5th day, you bring mouse, sorry, I don't have a scheme for that, but it's simple. On the 5th day, you bring the mouse to the same pool. This time, no cues. There is no even hidden platform. You just place the mouse in the swimming pool, in the tank, and the software tracks the time the mouse spends in the quadrant that formerly had the hidden platform. So in this case, we measure the memory. If the mouse has an intact memory or good memory, it will take a mouse less time to find the quadrant and spend more time in that quadrant because it thinks that the hidden platform should be around there. So in this task also, both groups performed similarly, and we concluded that ASD astrocyte chimeric mice exhibit no deficits in spatial learning or memory. So we found specific deficits in fear memory, and ASD astrocytes also induced repetitive behavior at the end of this behavioral experiments. So because there's bias, there's male bias in ASD prevalence, we also performed sex split analysis for the behavioral phenotypes we observed. Both female and male mice in both groups, control and ASD, performed similarly in marble burying test and fear memory test, suggesting that sex is not a main effect nor interaction factor that contributes to these phenotypes. So the only significance, the only factor to reach significance was ASD diagnosis. It was not sex. So because astrocytes positively regulate synaptic plasticity, especially LTP, and because LTP is the cellular basis for fear memories, which we observed. We observe fear memory phenotypes in ASD chimeric mice, and whenever there is a memory deficit, it's usually associated with a deficit in LTP. Since astrocytes positively regulate LTP, we were particularly interested in LTP measurements, long-term potentiation measurements in ASD chimeric mice. So LTP can be measured from acute brain slices, and for this experiment, we collaborated with the lab of Eric Colak at NYU. His former postdoc, Francesco, prepared these acute brain slices from our chimeric brains and induced LTP by electrical stimulation. So basically, you stimulate synapses to become stronger, and then you measure their potentiation. So upon stimulation, synapses in both CA1 synapses in both chimeric slices showed potentiation, but the potentiation was much lower in ASD chimeric slices compared to control chimeric slices. So this experiment showed that ASD astrocyte chimeric mice exhibit impaired hippocampal LTP, a phenotype that is actually very consistent with memory phenotype. To get more mechanistic insight into how ASD astrocytes induce plasticity deficits and behavioral deficits, we wanted to simulate the in vivo microenvironment by co-culturing our control and ASD astrocytes with with wild-type mouse hippocampal neurons. Because these two culture conditions closely mimic the cellular interactions in our chimeric brains. We then assessed both neuronal structure and structural properties and electrical properties, which I will show in the next slide. So here, first we addressed if ASD astrocytes induce any alterations in spine densities in healthy neurons. And in fact, healthy neurons, wild-type hippocampal neurons that were co-cultured with ASD astrocytes showed reduced spine densities when compared to neurons that were co-cultured with control astrocytes. Astrocytes are also known to regulate synaptic connectivity and especially synchronization of neurons in a network. Therefore, we also wanted to assess neuronal connectivity properties of healthy neurons when they are co-cultured with control versus ASD astrocytes. And for this, we used MEA, microelectrode arrays, and I'm sure you're familiar with these arrays, but microelectrode arrays basically measures action potentials in extracellular action potentials in a non-invasive way. You plate your cells, you culture your cells on a plate that has microscopic electrodes on the bottom, and the software can calculate or can record mean firing rate, synchronization, network bursts, etc. So we plated our co-cultures. So ASD here refers to ASD astrocytes co-cultured with healthy neurons, and control refers to control astrocytes co-cultured with healthy neurons, and none refers to only neurons, no human astrocytes. So we plated we cultured our co-cultures on these plates and measured mean firing rates and synchronization and network bursts. And ASD neurons that were co-cultured with ASD astrocytes showed reduced mean firing rate, reduced synchronization, and reduced network bursts. So we concluded that ASD astrocytes decrease neuronal network firing on primary hippocampal neurons when compared to control astrocytes. Yes, I mean, at this point, it was obvious that ASD astrocytes induce specific cognitive and behavioral deficits by influencing the structural and functional properties of neurons. So, so far, what I only showed you, so far I showed you the calcium deficit in astrocytes and inherent deficits, and I showed you phenotypes that ASD astrocytes cause, either in chimeric brains or in vitro in co-culture experiments. But I did not show you any experiment that linked this to each other. So therefore, we performed a rescue experiment and asked, does attenuation of calcium mobilization, in other words, does modulation of calcium activity in ASD astrocytes prevent dysfunction induced by these cells? And to do that, this slide can look busy, but don't worry about it. It's very simple. We basically wanted to lower the calcium responses in ASD astrocytes, and for that, we targeted IP3 receptors. IP3 receptors are receptors on the surface of endoplasmic reticulum, and they are responsible for calcium release to cytoplasm. And luckily, these receptors are only known to regulate calcium signaling and nothing else. So you are not disrupting anything in cells when you knock down these receptors. But our knockdown was also, I'm not showing here because it was too much as a validation experiment, but that we knocked down the IP3-1 and IP3 only like around 50% because we didn't want to eliminate the calcium response. We just wanted to modulate it, bring it to levels similar to control neurons. And we did the validation experiments. We used high throughput calcium mobilization assay. And here ASD none refers to ASD astrocytes with control virus, and ASD knockdown refers to ASD astrocytes with IP3 receptor knockdown, in other words, with modulated calcium. And ASD knockdown astrocytes showed reduced calcium response upon activation. And these others are more like controls, negative controls, or controls to test. Overall calcium levels were not affected, but only activation or response to stimulation was reduced in ASD knockdown astrocytes. So perfectly, we have now ASD astrocyte with exaggerated calcium response, and we have now ASD astrocytes with modulated, kind of normalized calcium response. And so we repeated the MEA experiment now with our ASD astrocytes with modulated calcium response, and those astrocytes with modulated calcium response displayed similar properties to control astrocytes in terms of mean firing rate, synchronization, and network bursts. We also transplanted these astrocytes with modulated calcium responses into mouse brain, and in chimeric brains, we assessed if modulation of ASD astrocytes prevents deficits that we formerly saw in peer memory. In fact, these mice that were transplanted with ASD astrocytes with modulated calcium activity performed similarly to control mice and did not show, when compared to control mice, did not show a contextual peer memory phenotype. So in summary, we found that ASD astrocytes induce repetitive behavior as well as deficits in memory and synaptic plasticity, which reflects changes in neuronal network dynamics. And our study actually identifies altered calcium signaling in ASD astrocytes as a mechanism that contributes to specific behavioral and neuronal deficits. So of course, there are a lot more to do in future because first of all, astrocytes are very heterogeneous and they're complex, and we even don't know, whether we were working, like whether our organoid astrocytes are just one population of astrocytes or mixed population of different astrocytes, but only a single cell RNA sequencing in engrafted mature astrocytes could answer that. And we didn't study the glia transmitters released by astrocytes and their impact on structural plasticity, that whether this exaggerated calcium signaling or response in ASD astrocytes cause release of glutamate or D-serine or ATP, we don't know. And another interesting, so we did not study motility or phagostic activity of these astrocytes because we showed that ASD astrocytes alter spine densities when they are co-cultured with neurons, but we don't know whether this is because the phagocytic activity of ASD astrocytes are altered or not. That would be an interesting future experiment. And finally, because we wanted to, in our in vitro experiments, we wanted to simulate the in vivo environment to complete, because the in vivo in vitro experiments completed each other in our study, we used mouse hippocampal neurons in our co-culture experiments. It would be interesting to see if ASD astrocytes, how ASD astrocytes actually influence human neurons, whether similar to mouse neurons, whether they will induce spine deficits or mean firing rate or synchronization phenotypes. We don't know that. So with that, I would like to thank my lab members and collaborators, a lot of collaborators, and I'd like to thank you for your attention, and I'm happy to take any questions.
So this is a lot of work. I mean, I'm quite amazed that you were able to move like from the in vitro system to this kind of a chimeric approach in even the co-culture with the astrocytes in mouse neurons. So you kind of covered it all, and I'm glad that you went from the molecular aspects to behavior. And that's one question for you. I mean, if I might start, I have plenty of questions here, but the first one is, so you transplant these astrocytes in P1 Just newborn animals?
P2, P3, P1 is too little, P2, P3.
Okay, and they're not rejected by the animal?
They're not, because we use immunocompromised mice. These are REC2 mice. So you do transplantations. The most successful transplantations are in immunocompromised mice. So these mice, they do not have T cells and B cells. Okay.
Okay, so they do not reject the cells, and then
yes, so they do not attack the transplanted cells. So they do not phagocyte the transplanted cells.
And I mean, I'm amazed that you see behavior changes upon transplantation. How many cells? I mean, first of all, did you transplant astrocytes, or these are kind of a glial progenitor cells?
Oh, yeah, I'm glad you asked that, because people maybe I should have been more clear. We actually, so because there is this pioneer study from Goldman Lab where they transplant A2B5 cells, more like progenitors, and they become both astrocytes and oligodendrocytes. Because we were interested in the role of astrocytes, we kind of we transplanted only the cells that showed ALDH1L1 and GFAP expression, so around passage 8 or passage 9. So compared especially to previous transplantation studies, our astrocytes, our cells were astrocytes. They were not progenitors anymore. They were like, kind of mature. But of course, they got maybe even further mature and got complex in the brain. But that kind of, I mean, there are advantages and disadvantages to this, because so they were mature. That's why in the adult brain, we don't see oligodendrocytes, we don't see microglia, we don't see other cell types. We only see astrocytes that survived from transplanted cells. But the numbers were not as high as the Goldman would get, because you will imagine that at progenitor stage, they proliferate more and they really invade the brain. So in our case, they didn't invade the brain, but they were more like targeted cells, and so they populated mostly cortex and hippocampus. They proliferated because we see millions of cells, but they did not really took over native astrocytes that I can say. So there were still native astrocytes there.
But the fact that they do proliferate, meaning that they are already committed to the astrocyte lineage, and obviously, I mean, you don't have the whole integration in the brain, but the spread that you show, I mean, it's quite good. I'm glad to see that. Yeah.
So yes, I mean, you transplant in 4 positions, but of course the brain expands too. So they proliferate, they migrate to a certain extent, and most importantly to us, that at the end, at least we can conclude that these are astrocytes, and astrocytes do this and astrocytes do that. But it's true that maybe if in terms of number, if they were more, maybe we couldn't see certain phenotypes because of the number. But we were still happy with what we have, because at least now we know that astrocytes, our study suggests that astrocyte can contribute to specific behavioral outcomes of ASD, also plasticity.
These are the socialization versus the repetitive behavior. It's amazing that we're able to distinguish those and connect.
Yeah, that was amazing. And I was thinking about it yesterday, because I was reading an abstract, a conference abstract, of how striatum is very important for social behavior. And I was thinking, is this the reason? Maybe because our astrocytes are mostly in cortex and hippocampus. I mean, is it that because, maybe if we transplant to striatum, if these astrocytes were in striatum, maybe we would see a social effect? Or it could be that sociability is something really to do with neurons. It's inherent to neurons, and astrocytes are nothing to do with that. So, like little by little with this kind of studies, we are, trying to understand, trying to dissect which cell is responsible for what.
That's fantastic. I got, similar questions here in the Q&A. Asking about the transplantation. I think people were curious about that. There is another question: if there is a dose response based on exogenous astrocytes with alteration of behavior.
those means like different, transplanting different numbers.
Yeah.
Well, I mean, I think our idea here that we would not even know what we would see. So, we transplanted a certain amount and we did not do any of those experiments. Yes, it's actually in the paper we also wrote in our future directions that it would be interesting to know how many astrocytes you would need to see a phenotype like this. Yeah. It's same even for in vitro experiments. We just used a fixed number of astrocytes in vitro for healthy cultures because that was our priority, where neurons would look healthy with a certain amount of astrocytes. But it could be interesting also for LTP studies, like how many astrocytes you need in order to see this effect. Yeah, these are all future, directions.
I think now you have a system and you can start exploring those questions. There is another one still related to the transplantation, which, by the way, I mean, it's fantastic. That's why people are asking about. Were cells injected in other brain regions, or all the injections are always in the same sites? Did you try different sites to see if anything happened?
Well, I mean, these injections are manual, but you can also use stereotactic. But we did 2 injections before BRCMA and right behind, in front of BRCMA and behind BRCMA, 4 injections in total. And again, so this is very critical, that we try to inject only in cortex, but I mean, the syringe is in your hands, and then you have a little pop in front of you.
I have to remind people that this is like early newborn days, right? I mean, it's not that you have
but I'm sure if again, so this is that's why I said my summary was only 2 lines, and then the future experiments were too long, and it was that was even the summary of the future experiment. So one could make it more automated and more precise. So I'm sure there would be cases that we hit striatum, we hit maybe other regions, but when we did quantification, which I didn't show you, that how many cells we did in the brain, what we saw consistently was the fact that the cells were in cortex and hippocampus mostly. So it's important also that always the same person does the transplantation. So, it's consistent. But again, I mean, I'm sure there were cases because, especially in the behavioral experiments, we didn't take out the brain and slice and examine each brain that, oh my God, where the exactly cells, where did the astrocytes go in this exact brain. So there could be cases that, we hit some other regions, but what we mostly saw in our hands when we did these injections, they were mostly in cortex and hippocampus because we tried to stay on the hippocampus, stay on the cortex, when you go with the needle.
Very nice. I have another question here regarding the genotypes of these autistic individuals. If you sequence them, if there is any specific gene, just a side note, I was also in the beginning very interested in idiopathic autism, but it's such a hard thing. I mean, you try to publish, the reviewers just beat you up.
Oh my God.
Most of my research now is based on single genes. So I think you are brave. And that's what we should do.
That's true. Because I'm also, believe me now, because they basically think that, these, it's very heterogeneous and then they are very different. So you cannot, study them. But I think that's wrong. And especially our proteomics data suggests that, because as heterogeneous as they are, they have to converge on certain common pathways because the phenotypes are similar. I mean, that it's still one single disease.
That's the same thing for Alzheimer's. Otherwise, nobody would study Alzheimer's. Right?
Exactly. So otherwise, I mean, you would not call it a single disease, right? Or certain symptoms are there. And that's why we were not surprised with proteomics analysis where we couldn't pinpoint a single gene. But like a lot of the very protein expression converged on calcium signaling, and it makes sense. So maybe a protein protein A is affected in one individual, protein B is affected in that individual. You would think they're very heterogeneous, but maybe those two proteins are serving on the same pathway. But yes, for the gene analysis, we did whole exome sequencing, and we collaborated with Betty Ross's group. They are a genetics lab, and so there were no any known CMVs or any known gene. So our lines were not associated with any known genetic factors of ASD. And when we picked our lines also, we tried to pick similar, for example, all of them had low IQ. They scored very similar, and all of them had ADOS scores, and they scored very similarly, at least, and they showed more or less similar phenotype. All of them show repetitive behavior. All of them show sociability deficits. So as heterogeneous as they are in terms of genetics, already in the selection process, you could see that common pathways should be altered in these individuals, because the phenotypes are very similar in terms of behavioral phenotypes were similar, low IQs and all. So we were careful with that they were comparable to each other.
On a related note, do you think that this is applicable to other members of the spectrum?
Yes, you mean Fragile X syndrome or Rett syndrome? If in terms of the astrocytes, then if you think astrocytes play a role, of course, why not? For FMRP, for example, FMRP is expressed in astrocytes too, and FMRP regulates mRNAs in astrocytes too. So yes, it could be.
Yeah, and I can say from Rett syndrome that the disease we work, it's quite important. Yeah.
Yeah.
All right. Another one. Where the iPSC's line donors approximately age matched? I don't know if...
Yes, at the time of, at least ASDs were controls. We tried to match them as much as possible, but the ASD lines, yes, by the time the sample was collected, they were all, very, very young age.
Okay. And I don't think that matters, right? Once you reprogram them, I'm assuming that, the age factor is gone. So
I think so. And it's different compared to other diseases, though, like with Alzheimer's, because people can show it at different stages. But with autism, it's usually, especially these days with the iPSC technology, if you know someone, the diagnosis is usually around the same age. So, then you find a lot of samples that were collected around the same age.
Yeah. No, that's amazing. I think that's these are the questions that we have. I would thank you again for the fantastic.
Thank you very much. It was my pleasure.
Tour de force here. Moving from the molecular biology to all behavior is.
But again, you can see we were very lucky. There is no way that we have expertise in all these techniques, right? We were very lucky to have Connor Liston next lab, and even luckier that there was a person in his lab who was experienced in measuring calcium activity in astrocytes. That was heaven for us. And Eric Kalan, he was very helpful, and his postdoc was fantastic, Francesco. So combining electrophysiology, molecular biology, and calcium imaging. Yeah, so we needed a lot of collaborators, and I'm very thankful.
Thank you again, Dr. Colak, for your talk. Evan, any last words?
I think you really reinforced also that impactful science really is a team sport, and I
absolutely,
think, and that's an important lesson for trainees in science, that we should not be siloed. And bye-bye, everyone. Stay well.
Thank you. Bye.
Thank you. Bye.